Welcome to SciKon Innovation. We're glad to show you a simulation on how our SciFlow fluid flow plate system can help researchers in pharmaceutical and toxicological industries to involve cell signaling reactions in new experiments and assays. Nowadays, many researchers in cell biology require investigation of cell signaling, a complex process with plenty of possibilities for new discovery. However, the current 96-well cell culture plates only allow observation of cell reactions under a constant concentration, and it is difficult or impossible to put more than one type of cells in the same culture chamber and observe how secreted molecules from one type of cells affect the other. Each of the 8 rows of the SciFlow plate has 10 culture chambers connected by microfluidic channels, allowing drug or toxin to be delivered in a similar way as in human body. It also allows secreted molecules from previous chambers to flow to the following chambers with incoming drug or toxin. This simulation video shows the movement and concentration of only the secreted molecules, shown in red, when all culture chambers have been loaded with the same kind of cells. Secreted molecules from one chamber are carried by the fluid flow to the next chamber, and the later chambers have a higher concentration of secret molecules. Adding it onto the flow and concentration simulation of incoming drug or toxin, shown in blue color in this simulation, it can be observed that the secreted molecule and incoming toxin merge into the purple color mixture as incoming reactant for cells in later wells. This increasing concentration of secreted molecules and decreasing concentration of incoming toxicant at one time spot across the chambers are two variations that may be utilized in designing experiments. In this simulation, one type of cells sit in the first 5 chambers and another type of cells sit in the last 5 chambers in the series. Their secreted molecules are shown in red and green respectively. The incoming drug or toxin is still shown in blue. SciFlow made observing cell signaling between different types of cells under incoming drug or toxin in an in vitro environment possible. In conclusion, SciFlow has made in vitro, in vivo benchtop multi-organ experiments possible, as molecules secreted by the cells are allowed to flow through the channels and reach other cells in following chambers. Different types of cells and tissues may exist in the same row, and researchers can observe cell signaling between them just as in human body. The concentrations of incoming drug or toxin changes with time and location, giving opportunities to exciting new experiment designs. 8 rows of chambers on one plate can give up to 80 samples and concentration data points at one time, significantly raising the effectiveness in surveying cells.